Hello, word group number 1, and we'll be presenting on horizontal gene transfer. My name is Rachel.
My name is Landon.
My name is Brooke.
My name's Carter.
My name is Paige.
And my name is Aaron.
Alrighty, to start off, I'll be doing the introduction. So, what a horizontal gene transfer is, it is the process where microorganisms acquire genes from other cells, and this is to adapt to varying environmental changes. And scientists often do this to intentionally move one gene from a donor to a recipient gene or to a recipient bacterial cell. A recombinant is like the mixture of both qualities of a donor and recipient. And there's three methods for horizontal gene transfer. The first one is going to be transformation, where an unclosed or naked DNA is then picked up or absorbed by a bacterial cell. The second one is going to be transduction, which is done by a bacteriophage, where it transfers the bacterial DNA from donor to recipient. And the last is conjunction, where DNA is transferred by contact between two different bacterial cells. And lastly, homologous recombination is the process where DNA of the donor is very similar in nucleotide sequence to the one of the recipient cell genome, and this is to entirely replace the DNA of the recipient with the donor's DNA.
Bacterial transformation is defined as the uptake of naked DNA by recipient cells. Naked DNA is free roaming in the cell's surroundings, which originates from previously lysed bacterial cells. Another source of naked DNA stems from a certain species of bacterial cells which release small fragments of DNA as a means of facilitating transformation. In order for a cell to successfully take up DNA to perform transformation, it must be competent. Competence is described as a psychological cellular state that allows them to take in naked DNA. Most bacteria will accept any form of naked DNA, but some species only accept DNA from related species. Some prokaryotic bacteria may become competent naturally or are always competent. The natural process only takes place under specific conditions, such as when there is a lack of nutrients. Most other bacteria do not become competent naturally, but can be facilitated in a lab setting to become competent by using certain compounds and conditions. This section focuses on the natural process. There are 4 steps to the process of natural transformation. The first part is DNA binding. Once the cell is competent, it expresses DNA binding proteins on the surface. These proteins recognize and bind to double-stranded DNA from the environment. Next is the DNA uptake. In most cases, one of the 2 strands of the double-stranded DNA is degraded by a nuclease as it is transported into the cell. The other single-stranded DNA is pulled into the cytoplasm. DNA uptake is mediated by a complex of proteins that form a transport channel through the cell membrane. DNA integration. Once inside the cell, the single-stranded DNA must integrate into the bacterial genome to be maintained and passed on to the daughter cells. This integration typically happens through a process called homologous recombination. If the incoming DNA sequences are similar with the regions to the bacterial chromosome, recombination proteins help align and incorporate the new DNA into the chromosomes. And once integrated, the new DNA may be transcribed and translated, allowing the new bacterium to express new traits. If the acquired DNA provides a beneficial trait, the bacteria have a survival advantage in its environment.
A second type of horizontal gene transfer is transduction. Transduction is the transfer of bacterial genes from a donor to a recipient cell by bacterial viruses. Bacterial viruses are more commonly referred to as bacteriophages or more simply phages. They contain genetic material which can either be RNA or DNA and a protein coat. There are generally 5 steps for a transduction to occur. The first is attachment. This is where the phage will attach to a specific receptor on the host cell. Next is genome entry. This is where the phage will release an enzyme called lysozyme to break down the host cell wall, and it will inject its nucleic acid inside. Nuclease is encoded by the phage genome, and it helps to break the bacterial DNA into smaller pieces. The third step is synthesis. This is where the phage will use bacterial cell enzymes such as the DNA polymerase to replicate its own nucleic acids and synthesize the proteins needed for the coat. Then assembly takes place, and the phage nucleic acids enter into the phage coat, and various components assemble to produce a complete phage particle. Lastly is release. In the phage particle will release lysozyme again to break down the host cell wall and exit, allowing the particles to attach to other bacterial cells to begin new cycles of infection. There are two types of transduction. The first is generalized. Generalized transduction is an error during the assembly of phage particles. During the assembly, a piece of bacterial DNA enters the phage protein coat and creates a transducing particle. It is referred to as a transducing particle and not a phage particle because it does not contain any phage DNA. The transducing particle is then able to attach to another bacterial cell and then inject the bacterial DNA it contains. The bacterial DNA may integrate into the recipient's chromosome by homologous recombination. Any gene of the donor cell can be transferred. This is why this mechanism is called generalized transduction. Now we will look at the second type. The second type is specialized transduction, which is an excision error made by a temperate phage during the transition from a lysogenic cycle to a lytic cycle. At the start of a lysogenic cycle, a temperate phage first injects its DNA into a bacterial host, and the phage DNA integrates into a host chromosome, creating a prophage. For various reasons, the lytic cycle can be activated, and the prophage becomes a cyst from the host chromosome, meaning that it is cut out. Rather than the prophage being a cyst precisely, a mistake is made and a short piece of bacterial DNA on one side of the phage DNA is taken. The bacterial DNA on one side of the phage is taken, so now the excess DNA will contain both phage and bacterial DNA. Once released, the defective phage injects the DNA it contains into a new bacterial cell. Compared to generalized transduction, which can transfer any gene, only bacterial genes that are adjacent to the integrated phage DNA can be taken in specialized transduction. Transduction is important because it helps increase genetic variation in bacterial populations, and it creates the possibility for gene therapy. Many researchers are currently studying how viral vectors can be used to introduce genetic material into target cells to potentially treat diseases that were previously seen as incurable.
All right, so I did mine on conjugation, and the first one is the most common one, which is plasmid transfer. Conjugation is the process of transferring DNA between two bacterial cells. During the plasmid transfer, the F plasmid is transferred using the F pilus or the sex pilus. The bacteria containing the F pilus is notated as F positive, or F plus, while the bacteria without an F pilus is notated as F negative. The plasmids, being replicons, refers to their ability to replicate in a cell without integration of the cell's receptor chromosome. Essentially, they are able to replicate their DNA without help from the cell that they have transferred into. The spread of antibiotic resistance is a consequence of plasmid transfer because once the transfer of plasmids from one bacteria to the other is complete, the cells are both F positive. All right, and then the second one is a less common process of chromosome transfer. In these high frequency of recombination cells, the F plasmid integrates into the bacterial chromosome. The DNA that enters the recipient cell is not a replicon, which means it is unable to replicate by itself. If the DNA does not integrate with the recipient chromosome, it will be degraded and will remain an F negative cell. HFR cells can reverse back to F positive because the integration process is reversible. An F prime cell can occur when there is an excision error that transfers chromosomal fragments along with the original DNA.
Genome variability led to the concept of pangenome. Pangenome is the total amount of genes encoded by the different strains of a species, and it consists of the core genome, the accessory genome, and unique genes. The core genome is common to every strain of the species. The accessory genome is present in some but not all strains, and unique genes are found in only one strain of the species. Mobile genetic elements contribute to accessory genomes. Mobile genetic elements, or MGEs, can move from one DNA molecule to another. The total set of MGEs in a genome is called the mobilome. Examples of MGEs are plasmids, transposable elements, genomic islands, and phage DNA. Plasmids are common and have circular double-stranded DNA. Our plasmids encode to be resistant to antibiotics. Transposable elements are pieces of DNA that can move to a different location within the cell's genome, and an example of this is an insertion sequence and transponsons. Genomic islands are large DNA segments in the cell's genome that originated in another species. And phages can insert their DNA into the host cell's genome, which then gets replicated and passed on.
So because bacteria are mostly affected by pathologies that are DNA and RNA based, their immune systems are also DNA and RNA based. And the most basic system that it has, these systems pose a hurdle to transduction, conjugation, and transformation of the genome from outside elements. The most common and the simplest mechanism is the restriction modification systems. And related bacteria will have similar restriction modification systems, meaning the enzymes they use for it are the same. And different bacteria that are not related are going to have different enzymes. And these systems are composed of two enzymes, an endonuclease and a methyltransferase. The endonuclease does the restriction part. What it does is it chops up unrecognized or foreign DNA as it enters the cell. And the modification enzyme will add a methyl group, or it will methylate the bacteria's own DNA to make it immune to or incompatible with the active site on the endonuclease enzyme. So this system is not perfect. Sometimes mistakes are made. Sometimes it doesn't methylate its own DNA properly and accidentally cuts itself. Sometimes it accidentally will methylate foreign DNA, and then the foreign DNA is immune to its own restriction enzyme. Those are rare and they can be corrected, but it does happen sometimes. In addition, the other system available is the CRISPR systems. CRISPR systems are more complicated. It stands for clustered, regularly interspaced, short palindromic repeats. And what happens is foreign DNA from a bacteriophage gets injected, and then the bacteria will save a piece of that DNA or RNA, and it will incorporate it into its own genome as a spacer in the CRISPR gene, so that if a repeat infection from the same type of bacteriophage occurs, the bacteria will be able to recognize it as a repeat infection and directly attack and more quickly stop that kind of infection. It uses the spacers to make match cRNA or mature CRISPR RNA, which is a complementary single strand to the infectious DNA. And then it will use Cas proteins or CRISPR-associated proteins to destroy that recognized repeat infection DNA or RNA. There's a lot of different types of CRISPR systems that exist. Again, this is a way to recognize if bacteria might be related or not. If they use the same kind of CRISPR system, they're organized into class 1 and class 2. Class 2 is the simpler system. Class 2 means that it uses a single enzyme to attack recognized infectious nucleotide sequences. One enzyme usually paired with tracer RNA, trans CRISPR RNA, which is what the Cas protein uses to recognize the mat cRNA and find it once it has attached to foreign DNA. class 1 types use multiple enzymes. So instead of a single enzyme with a tracer RNA, it's multiple enzymes, possibly with multiple tracer RNAs. And bacteria might be related if they use one or more of the same proteins or enzymes in that sequence. So they might have one similar protein or enzyme out of 3, or they might have all 3 similar. They might have 6 enzymes that are all used in this immune response to destroy the foreign repeat infections. The most common researched CRISPR type is class 2, type 2, which uses the Cas9 enzyme. This CRISPR system is currently being used in test models to treat various diseases, such as sickle cell anemia, with some success. It's also being used to research different types of virus-mediated mutation to make custom bacteria for other medicinal or production purposes. This leads us to the conclusion of our presentation with our Christian worldview, and we've selected the verse Colossians 1:16 and 17, for in him all things were created, things in heaven and on earth, visible and invisible, whether thrones or powers or rulers or authorities. All things have been created through him and for him. He is before all things, and in him all things hold together. And we see the fact that we can use these systems for medicinal purposes. We can recognize these patterns as evidence that God has ordered things that we can see and ordered the things that we cannot see, including microscopic processes that occur within these bacteria. And these things can work together for our good because He is good.
And here are our references. Thank you for listening.